graded all of your midterm exams, so you should be able to see your grades there. In general, I think it was a good response, except very few people. So one thing when I asked about the flow diagram, so some people explain it in a paragraph, okay, how this thing will be fabricated, but the idea was to make diagrams, at least make blocks and say, okay, this step will be done first and this is second. Ideally, it should be the structure itself. Let's say you take a small wafer and then you draw the silicon dioxide layer, or if you're doing a lithography, draw that. So that was expected. So, but anyways, I think there was just a feedback. I already gave feedback on your paper, so you should be able to see it. But that's one thing which many people didn't do it. So I didn't take that into much consideration if you have explained your answer properly. If you have not drawn figures, still I didn't get directed to a lot of marks, maybe 1 or 2, so very little. So, but in general, I think people did well. I think minimum score is about 100 or something, out of 150. So I think it's a good average. Would be about 120 or 30, something around that. Okay, so and now you can see where you stand in terms of your overall grade, right? So everything is very clear. Just a group research proposal, if you do well, I think you can get a good score on that. But the final exam, that will be the last thing. So you can actually calculate your grade right away, where you fall right now, based on 60% marks, right? So far we have covered 60. No, homeworks are still, there is another homework due, and there might be another homework also, fourth one. So I think we have covered about 50% of the grades. And if you are getting, but for A, I think you have to get 90 or above. That's, I think, in a syllabus, so you can see it. Make sure keep working hard for people who are doing well. If people who didn't do very good so far, I think you should start working hard, because still there is a 50% left. Make sure you do good effort on your group research proposal. Make sure you prepare for your final exam. So if you did well if you will do well in both of that, so you can still make up to a very good grade. Okay. So last time we were discussing about a PCR, and in a PCR, so you will tell me now. So PCR, although it's a very good technology and it's the most standard method used for DNA or RNA quantification, gene expression detection, gene detection, and then also in a lot of infectious diseases. So, but there are some limitations that the reagents are a little bit expensive, and then also it requires a very labor-intensive process. It takes a longer time. It requires a trained technician. So this is good if we have labs. For example, in the developed world, we have labs everywhere, so we can do that easily. But sometimes we want to do it rapidly at home or maybe at a point of care settings, and that is very important when we are talking about some underdeveloped or developing world. So they don't have a very high-tech labs or equipment all the places, and they don't have actually people, trained technicians who can perform the assay. And the equipment itself is expensive, so about at least 10K onwards. So there are efforts to actually find out alternatives, how we can still detect the disease, for example, and without by using the same PCR process or maybe some other process. And one effort was this paper. You can look at this paper. In this one, they try to actually design a microfluidic device and try to do all the PCR steps on this chip automatically. And you can see this device, there are different chambers. There is also PCBs, means there are electrodes at different places which are turning on and off one device at one time. They also have pumps there which can pump liquids from one place to other place. So these are small pumps. They have valves. They also need a heater there. You know why need a heater? Anyone? Why we need a heater here in this chip? To denature the DNA and
heating the chip
for
maintaining a certain temperature.
For one is denaturation, right? So to make a single strand, that's one thing. And that is that the only purpose of heating, or is there anything else where we need heating also? Denaturation, annealing, primer binding, that's an annealing process which requires about 60 degrees. Then there is a third step, elongation, which 72 degrees. So there are 3 different temperatures which are required. So temperature is very important, otherwise you cannot amplify things. So all what we have covered so far on a PCR is the same thing. So you have to perform all of that on device, like you have to heat up certain chambers very quickly. But now in the microfluidics, the volumes will be very small. So the idea is that you can quickly ramp up the temperature and you can quickly lower the temperature because the volume is small. And then it has to be super sealed, the device itself, because if it's not sealed well, then the liquid or sample can evaporate very quickly because it's a small volume. But they have shown, and you can look at the paper for more information, that but I didn't see myself anything in a commercial domain like microfluidic device which can do PCR on chip, and I don't to my understanding there's nothing out there what people are using. So these devices are good, but there are some limitations also. They are not very reliable in terms of their accuracy. Sometimes they work well in a research environment, they work well, but when people who have not designed these devices, when they work on these things, these devices are not that reliable. And for clinical testing, it's very important that these devices should be reliable. So those are limitations which can be worked out, and people are working on it. Still, it's a very hot idea. So I searched on Scholar PCR on chip, PCR chip, that keyword, I think day before yesterday, and there were 637,000 hits. And when I searched it in 2008, I believe, or you can still search it and select before 2008, and there were only 56,000 hits. So from there you can see this is a significant area of interest, and a lot of work has been done in this area recently, and still people are working on that area, how we can automate the disease detection, how we can put PCR on devices. So this is a very important area. Maybe it may give you some thoughts about maybe proposal also. It's just one example. It doesn't mean that you have to choose from this. You have to choose something about PCR, but that's just a one topic which you can think about choosing. Like what else we can do to improve how PCR is done, for example, and what are the limitations of these devices, or like that. So anyways, so for DNA sequencing, we already went through previously. I think we discussed that. So briefly, for DNA sequencing or DNA analysis, we use gels. There is a gel, it's called agarose. And you mix it in a buffer, and then you can make up a gel, and then you put a DNA at one side, you apply voltage, and because of a DNA charge it moves to the other side, and then the movement is dependent on the length of the DNA strand. So you can end up actually getting the DNA isolated based on their sizes, and that can give you some information that what kind of DNA is present there. And you also have a ladder which is control, which can tells you okay, where, if there's 200 base pair, 300 base pair, 400 base pair, so where should that stand? So what can you do is I can show you. one image just for understanding how the gel looks like. Okay, these are real results. Okay, oh no, these are not real because they are super clean. This one maybe, for example. Okay, so I don't know what is there, but this is how the results normally look like. So there is a ladder. Okay, so this is the ladder. So in ladder it tells you, we already know what kind of bands we are expecting. So you put a DNA here, you apply voltage on this side and that side, so negative on this side, positive on that side. So DNA moves towards the positive, and they will, based on the size, they will, so same size DNA strands will come up to same place because they move at a certain speed. And then the ones which are shorter, they can move faster and longer. Once they are big lengthy ones, let's say 50 kilo base pair means 50,000 bases long, and this is a 0.1 kb, which is 100 base pair long, like 100 bases long. So in that way you can have that we call it a ladder. And now let's say if something shows up in this case, so this is a control where you see that most of the DNA in the control was a lengthy one, so it's a 50 kilo base pair. But in here, in your real sample, we see that there is something there in that area, although it's not present in the form of a bands, but still we see something there. But so this is maybe a better thing to explain, better image. So this is ladder again, and now this is the first sample, second sample. So you see that in first sample we only have maybe 300 base pair long DNA. In this sample we have 2000 base pair long DNA, so we can determine the length of the DNA, what is there. Let's say if you are amplifying a certain segment of our RNA for infectious disease, so if there is amplification, you will see that segment very clearly at certain location. So let's come back to gel so that's called the gel electrophorese. It's a very straightforward process. And then we also discuss about the DNA sequencing briefly, and then I also showed you a video that how the sequencing is done. And then you can watch in the previous lectures. I gave one example. I think there were 4 or 5 bases, and then we have there was a segment which was 4 or 5 base pair long, and then we have some freely available bases. And then we have freely available bases, and then we also have some bases which have some fluorescent dyes attached with it. And then some of the bases during elongation process, the elongation will stop at a certain point where there is a base with the fluorescent dye, and so in that way we have a random length DNA structures where at certain places it will be stopping at 2, at certain places it will be stopping at third base, and then we will put it in a gel, and then they will arrange it based on their sizes. Let's say if there is a 2 base pair long, 3 base pair long, 4 base pair long, so they will arrange it in a gel, and they will have different fluorescent dyes, so you can put a laser there and let them flow through your channel, and you can actually find out whether it's A or G or C, because they will be arranged accordingly. And you know the dye color, if it is a red or green or yellow, so based on that you can decide, or blue. But there are other, so I will not be going into much detail. I'm not explaining it to you once again because it's already discussed and it's a recorded lecture, so you can always watch that. So that's why I'm just skipping through it very quickly. So in DNA sequencing, but that's the standard method, how they do it. So basically, in order to sequence even a 100 base pair, it takes much longer time to do the process. And now then, because 100 you have to generate random sequences, run a gel, and then look at what you have, and then maybe you have to repeat the experiment to make sure that what you're getting is correct. So now there is significant interest in developing technologies to improve DNA sequencing. When we say improve, means to make it fast, maybe high throughput. Let's say if we want to sequence a whole genome, which is 3 billion base pair long. So human genome. So if 3 billion base pair, if we can do only 100 or 200 bases at a time in a gel by using Sanger chemistry, I don't know how much time it will take. It may take years to just sequence the one human DNA. So, but for new clinical information and bioinformatics, people want to see the whole pattern of genes in someone's genome, and that's a very important area of research nowadays. And people looking into genetics more deeply, how they're involved and how we can improve it. So for that, we need to improve also sequencing technology. So from sequencing, what does it mean? It means that we want to know the sequence of DNA. For PCR, does it provide you that information? The sequence of the DNA or RNA? It doesn't provide you that. We already know that what's the sequence. And then we design primers accordingly, and we just amplify some segment, and we just get the answer whether that segment is present or not present. Okay? But in this case, why camera is now looking at you?
You should notify, because otherwise maybe they are recording that.
Yeah, because I just,
They are using that camera instead of this.
Yes, so because I just saw there's something changed, and I looked at it, and it was the student, so I don't know how to notify. Okay, let me see.
You will read the video, right?
Yeah, we have to change our position.
Behave good. Behave well. They're working.
We can notify them. The office is over there.
Yeah, it's right.
Oh, they're there? So you
Yeah,
can you
they're
please?
right here. You know where.
This is fine.
Smile. Keep smiling. There it goes.
Oh, it got back. Yeah, can you please call Kabir?
Maybe. All right. There.
I think it was because of you. So when you left, it again switched back to me.
No, it will switch.
Yeah, we'll see. If the camera knows, if it has a sensor that Kabir is back, then it might switch. Okay, so in DNA sequencing, there is significant effort. So it's a very important area. So we want to develop technologies which can help to sequence the DNA. DNA is very small in terms of a size, so we cannot really see with a microscope that what is a sequence. So when I was doing my PhD, my professor once asked me, okay, think about it, how we can improve the sequencing, and I put a lot of effort thinking about it, but nothing came to my mind. I was thinking about, okay, maybe we can attach some dyes with the A and C's on the DNA itself, and then we can image it maybe with the fluorescent microscope or do something around it. But then again, if the bases are just 0.34 nanometer apart, let's say it's very difficult to approach each base with the fluorescent dye, let's say, but if we approach it single base, then the fluorescence, how to sense, because if there is a 0.4 nanometer difference, so one will be green, one will be blue, for example, so you will see a lot of background noise. So it may be blue here, and then green, and then another blue, right? So the fluorescent signal which you will be getting, you don't know whether it's coming from this side or that side because it's very small. So I could not make up some design around it, although still I think about it sometimes, that how we can improve it. If something comes to your mind, I think it'll be very important and interesting, because sometimes anything can actually come to your mind and it can solve the problem, like how we can solve it, what technology we can use it to sequence. So there are a couple of technologies which are under development and still developed, like Nanopore, we discussed about it, right? So that's a very innovative approach. You will see thousands of papers on that. So it's a very simple idea that you put a pore and let the DNA pass through and see if you can get a signal. So think about it, okay? If anything comes to mind, you're more than happy to discuss with me. So there are other technologies which have been developed very recently, and we'll cover 2 or 3 next generation sequencing technology. They are called next generation sequencing. The first one is from Illumina. It's a company. If you're looking for a job in a if you're an engineer or biochemist and looking for a job in this domain, in bioengineering, I think this is a good company. They do a lot of hiring. Nowadays, still there will be some position available if you look at their website. So this is one good example where they mixed microfluidics and some biology all together to make up something which is really interesting and useful. And now they have now commercialized systems. A lot of people are using Illumina nowadays, and it has significantly improved the sequencing. So you have watched the video of the Human Genome Project, right? So they want to sequence the full human genome. So if Illumina was available at that time, so they would finish the whole project maybe in a year or maybe much faster. It took them about 10 years or something, so they can maybe do it now. You can do it in a couple of days, the whole genome. So it's so fast. But still, it's a complicated process. I'll let you know in a minute how it works. So how it works, let's say we have a long DNA, okay? So do you remember what they were doing in that video? In a sense, how they were sequencing it? So they were actually breaking it in small chunks of DNA because they cannot work on a full DNA at one time. So you have to break it into small chunks, about 100 to 200 base pairs. And so DNA will break very randomly. So you don't work only in one piece. You have maybe 1,000 or 10,000 different copies of the same DNA, and then you break them randomly. And once you break them, so some will be longer, some will be shorter, right? Because you are breaking the DNA randomly. So it can be chemical breakage, or it can be mechanical breakage also. So, and then they were selecting only those pieces which are about the same length, about 200 or 100 base pair. Okay, so once you select those pieces, and then they were sequencing those, and then later on, because 200 base pair pieces, and then later on by using computer tools and algorithms trying to rebuild from where this piece might come, because there might be some sequences from this region which is 200 base pair, and other maybe from this sequence, and then look at, okay, if there are similar sequences, so then they will merge these two together. They know that, okay, this is the full length.
Okay, great.
In this case, the Illumina, they use a sonication, just sonicate it, and then it will randomly break at different places. So they might have optimized the protocol, like how long.
They break it randomly?
Randomly, yes. Randomly, because we don't know the sequence.
Yeah, because you said mechanically, but...
Yeah, sonication is mechanical.
Yeah, but it's random.
It's random, yeah. It can be any place.
Yeah.
So, and then what they do is, because how their platform works, then they select only certain lengths of DNA. How you can select certain lengths of DNA from after breakage? Electrophoresis gels. Put it in a gel, run it, and see where it goes, what kind of bands you get. And let's say if you are interested in 200 base pair only, so you just cut that segment of the gel. So you can cut the gel, and then you recover the DNA from there, and that will be only 200 base pair long. And that 200 base pair long, because if you have thousands of DNA, so that might cover the whole sequence, because then the 200 can be from different segments, right? It's not from the same place. It can be from different places, 200. So once you have the DNA fragments, after running a gel, you isolate it. Now it's very clean. And now they attach actually adapters. So they call it adapter. It's actually again a DNA. So one is green, one is purple. One is pink, one is purple. So they are two different. It shows you different types. Okay, this is what they are designing. So they already know what's the sequence of these adapters. Okay, so with the ligation, they attach these adapters to one adapter of one color one side, and then the other with the other side. We'll not go into detail how exactly they do it. I just want to give you overview of how they perform the full assay. So once you attach the adapters, one color on one side and the other on the other side, now this segment will look like this, right? So next step, then they attach the DNA on the surface. So they have a surface, and it's actually a microfluidic device, maybe larger size, I don't know exact size, but it's a device which has millions of already designed, already immobilized, so they have millions of places where this DNA with the adapter can go and bind. So they make it single strand and with the denaturation, and then they let them bind on the surface. So you will see that these DNAs will randomly bind in many different places. So, and they don't know where one is binding or the other is binding. So they will bind randomly. But now you have millions of spots where they can bind. And they can bind very close to one another. They can bind a little bit far from one another, right? All these fragments. But we can control to some extent how close they can bind based on their concentration. So if they are present high concentration, they will bind very close in general, right? But if they are lower, so then they will be. So we can optimize the concentration and make sure that they are not very close. So once they bind, and we already know what's the sequence, like the starting sequence. We know the pink and we also know the purple. But we don't know what's in there, the sequence which we want to sequence, the part which we want to sequence. So then, if you look at the bottom surface, I don't know whether it's very clear or not. So on the bottom surface, you have some places where you have pink and purple already, right? Many different places. And then let's say if we take this example, so this pink will go and bind with the pink somewhere. Okay, that's how the binding was happening. So, and then this purple will then be binding. It will be bending because it moves. It's a DNA. It's not very long. 200 base pair is how long? It's very little. 200 into 0.34. No, it should be less than that. So 10 bases is
68.
So 10 bases is 3.14, and this is 200. So 10 to 20 into 60 something. 68. So 68 is very small in reality. So it bends, and DNA doesn't stay stiff. It moves, and in water, and then it bends. And now, let's say this example, this pink was attaching with the pink, right? And then now this is purple. So if it finds purple nearby, it will bind with it because purple is complementary, okay, to each other. So both purple doesn't mean that's the same sequence. It's actually complementary to each other, and that's why they're double-stranded. So then it will bind and make a bridge structure like this, right? And they make it, so if DNA is very long, for example, if you use, let's say, 2000 base pair long, then there are good chances that it will not bind and it will not make bridge. So that's why, again, in this case, you cannot sequence long DNA at once type. They have to set up, I think they can sequence about 200 or 300 base pair at one place. But now in this case, it's not only those 200 base pairs. There are 200 at this place, 200 at that place. They're in same chip, a small chip. And you have a million places, so they will bind different places. They will make bridge different places, and each segment is different. It may be same, but in that way you can sequence actually million different places instead of one place. And let's see how they sequence it. Still, right now it's just bound there, and now it make a bridge. So first they generate a library. They call it a library generation, but in reality they just amplify the DNA and make a cluster of it. Now let's see how they do it. So now we already know that it's bound there, and we already know that what would be the sequence of these purple and pink ones. So then we can actually design a primer based on this sequence which we know, and then we let the primer to actually amplify. We call it a bridge amplification. It's the same amplification like in a PCR you have a polymerase and then you have bases available, so it will amplify and make a double strand. Right? This is how it works in a PCR. So this is, again, you attach a primer, so make a double strand, and then you stop there. So once you stop there, you heat it up to 90 degrees again, 95. So then it will open up that bridge, because 95 double strand DNA will become denatured and become single stranded. So now instead of a one strand, then you have a two strands. Correct? So now you have a two strands. If you again, they will again bind with the other part, and if you repeat that process, amplification and denaturation, amplification, denaturation. So it's the same kind of sequence. You will have many of them, and every time it get doubles, right? So maybe in 30 cycles, you can have 2 raised by 30 different strands, but they are of the same type. So you are just amplifying that sequence, and now you will see these clusters. They call it a library. at different places on your chip where it differs initially the DNA was bound, and the cluster will look like this, for example. Okay, now you have one cluster which has the same type of unknown sequence. You have another cluster which is a different type of unknown sequence, another cluster, but we don't know still what's in there. Okay, but now we are trying to making cluster, and you will see in a minute then why we need a cluster. Now let's say we have a cluster, everything is there. Now let's say we attach a polymerase as before, and this time we don't let it make a complementary. This time we only flow one base. Let's say which base? A type. Okay? So what will happen? If we flow A type, only one type of base, it will only goes and binds to the clusters where there is a T. Where there is a T. If there is a G's and other bases, it will not bind there. And then in this case, all the bases are fluorescently tagged. Blue, red, green, green, red, and yellow. So now let's say we flow A, so red will go and bind only those places where there is a T. So then you stop the process after one base. So you only amplify the one base, and then you look at, and there's a small chip, so you then look at with the fluorescent dyes very quickly. Fluorescent imaging is not that difficult. And now think about, instead of a cluster, if we only have a one strand, will you see any fluorescence from there? Even if the single strand of single base will go and attach there, you might not see the fluorescence from just the one molecule of the fluorescent dye. But once you have a cluster, because all of them are of the same type, right? So this A, let's say if this is a A here at this point, so A will bind to almost most of them to the place where it has to be. So then this cluster will shine very rapidly, because in cluster you have a million different fragments, and then you would be able to see that. So what they do is then they shine it with the fluorescent with the laser, all four colors, and they see where it's shining, which place.
Why the same type gathered together?
Because when they make a bridge amplification, once you make a different, like first of all, it's randomly arranged, okay?
Yeah, they're random.
Randomly, different places. But now the bridge can only be very nearby, because it's very small, right? So once it's make a bridge structure, it cannot make bridge far. It will only be making bridge very nearby. So you amplify it, now there are two strands. Now those two strands will also make a bridge very nearby. So in that way, you keep them amplifying, and then it will be very localized. Okay? So then you look under the microscope or the fluorescent reader, and you can quickly identify wherever is the red. So when there is a red, you know that at this place we have T, right? Sometimes there can be two T's there, so it means the A will bind to both of them. So, but we still get a red. But this time, then the fluorescence would be higher than the one T case. So you can still identify that if there was only one T or two T, there can be three. If there are 10 T's or 9 T's, then it will become difficult. So there are chances for error, right? So if there is a sequence where there are 10 T's and you want to sequence it, if you put an A, it will bind everywhere. So then it will be hard to identify really, I don't know, if there are 9 or 10, right? But so these are some limitations, but still it's fine, because we don't see similar bases. We don't see a very long sequence of single bases. They are mostly AT, TT, 2 or 3 most of the time. So we can sequence it. Now you have to remove that dye, fluorescent dye, for the next base, because if you don't remove it, then now let's say the second we push the green dye, so it go bind with the place where there is a green. It may be something where already red was attached, or it can be something new place which start with the T, so A, right? So and then green will bind different places. You read it, then again C, and then G. But now again you will flow A. So if the previous base is already there and which has a fluorescence, then you push another A, then you don't know whether it's the same A from a previous. So it means in this process, you have to remove the dye after each step. So once you push A with the dye, you read it, and then you bleach out with the chemical, remove the dye from the base, and then push a second base and then third base. So one time one base, but every time you have to do the washing step so that the dye is removed. So in that way you can actually sequence, but in this way you can sequence a very long DNA. This process is, and they are selling a lot of these equipments, and now it's a very successful company, and I've seen it progressing, and so they're doing very well commercially. And now many places where people do sequencing, and they use Illumina systems, and it's a very recent development. So, and you can think about the idea itself is not that complicated, right? So a lot of people will know that we can use dyes, this and that, but how to do it and how to sense it, that was a trick. And if you have an idea, you can think about doing it. Now you can build actually all this by yourself. We don't have to do it on the whole chip. When you are developing an idea, if we show that 4 or 5 places that we can sequence the DNA like this way, the power is that now you can have maybe a million different spots on a chip, which you can sequence all of them all together instead of just one 200 base pair. That's the power. So you can ideally you can sequence very long DNA, and then you again that algorithm and attaching them back to where they might have came from. That's still and that's still an issue with how people are doing it before. So then and there's still interest in nanopore technologies where we may be able to sequence long DNA altogether. But I think that's still very low cost. $1,000 you can maybe able to sequence very long genome DNA. So it's not expensive as compared to how it was before, and it's very fast. There is another technology which is also very recent development. Little bit similar, but their sensing mechanism is different. So now you'll see that how the transistors and our devices and sensors can be used in a lot of applications in medicine. And sequencing is one example. So in this case, you will see that this device is called the Torrent Technology. And this is how the device looks like in reality. So this is just a chip, like a computer chip, right? So in a computer chip, you know already that we can have millions of transistors in a small area. And that's the power of how computer chips are built, right? So you can have millions of transistors in a small area, and based on lithography and e-beam lithography and all that, what we have covered. So that's called CMOS chip with the transistors. From wafer, we can cut many of them, right? Okay, so how we can use those for applications? So what happens in this case, and this is also a new technology. They might also have a lot of jobs nowadays. This technology is now, I think, owned by Life Technologies. It's a really big company in biotechnology, and they have a lot of other products. So this was developed by another professor, but they, I think, bought the patent and licensed the patent. I think now they are selling and developing it. But this is very recent, so I don't know whether they started selling their commercialized product, or is it still need to be finalized, but it's very recent. You can look at it. So in this case, how the chip works. So what they do is, in this case, they take microparticles, small glass beads. So let's look at the beads first on paper. Maybe it may be easier to explain there. So let's say we have a device, and then it has the sensors at the bottom, okay? So which we are not showing it here. I'm just showing the top side. And then this small chip has thousands of, okay, they are same size. Thousands of different holes or valves. They are same size, okay? And there are thousands of that valves, and we can easily make it with the lithography, right? So if you want to make a valve, you can use a resist, or maybe you can make these valves also in silicon dioxide, and you know that how to do it. And once we have many wells of the same size, and they will be very same size. Then you take actually some beads, which are glass beads. And now, and there are many of them. So the first step is similar. You take a long DNA, you break it down into small chunks, isolate only the small chunks, and then now you attach the fragments which you isolated on these beads. Okay, so ideally we want these beads to have only one fragment. We cannot really control it because you're mixing the DNA with the beads. So if you have less number of beads as compared to DNA fragments, right? So what you'll be ended up, you might have more than one fragment on each bead, right? Which is not desired. This is not what we want. We want at max one fragment on a bead. So how we can do it? We can have maybe more number of beads than required, and then let it incubate. So in that case, there are good chances. We can find also the probability number of beads and see, and we can also find out what is the probability of getting only one, theoretically one fragment on one bead, or maybe more than one fragment. So if in general, qualitatively, if we have more number of beads, then the chances that two fragments will bind on one bead will be lesser. Okay, so they optimize these settings. So the goal is actually to have most of the beads only one fragment. Maybe one bead in thousands have two fragments, or maybe there are quite a few beads which doesn't have any fragments. So that will be, so it's not 100% that each bead will have one fragment. So now you take the beads, which are their size are slightly smaller than the well size. Then they flow these beads on the surface at high concentration. So what happens that each well will end up getting these beads inside. And well is designed based on the size such that it can only hold one bead inside, not more than one. Okay, and this has to be done in a microfluidic channel where you fix the height, and then you let the beads pass through, so they will fall into the well. If it is a larger channel, then it will be harder to do it. So then you have these beads there, and then they have one fragment only, so they will just stuck there because of the well structure. There is no chemical bonding; it's just a mechanical. They're stuck there because bead is circular, so you make a pit like that, and then it will fall into pit. If you push it very hard, then they might get off from there, but you have to optimize those flow settings. Now you have one bead. In each bead we have one fragment of DNA. So again, in this case, they also amplified to make a library. And the amplification, as before, you have polymerase, and they will come and they will start amplifying everything. What is they designed? Okay, let me see how, what I'm missing here. How they make a library. So I don't remember whether they attach some adapters like before, so that you need to know some sequence to amplify the DNA. It can be, so I think you need to know some sequence, so it means that you need to have some adapter there also with the beads at this segment where this primer is binding. You need to have some adapter there, or once your beads are there, you attach some adapters afterwards with it, so that you need to know some sequence there, which I don't know exactly. I forgot to look into it. So it should be some sequence there which you know, and then you actually amplify it. So in this case, you'll be amplifying on the bead surface in this well. So now instead of one, you will have many of those fragments, okay, in each place. So each bead will have after 30-40 cycles millions of the same type of DNA on the bead in the same well attached with the bead. So what you do after that, now we can come back to our PowerPoint. So after that, once you have now, this is how the chip looks like, okay? You have wells, there is a bead. This is one bead, I'm just showing you one bead. And the yellow is the one fragment which you amplified, and they're sitting in one each well. And this is another image to show you. So what happens now? You flow one base. In this case, we don't have to worry about. This is it's a label-free technology. It doesn't require any fluorescent tags as required previously. So it means it doesn't also require that washing step, which is used to isolate, to remove the dye. So in this case, what's the sensing mechanism? That let's say now you flow A. Okay, when you flow A, it will bind with a T somewhere. When there is a T at the starting point, right? And you have a polymerase there. So if there is a, it will bind with a T. So if there is a T, whenever A binds with the T or G binds with the C, in reality, it binds. There is a phosphate group, okay, and which is a sugar group, which is attached with the one side at 3' and other side is 5', right? So where it's attached with the 3', the 5', then 3' end is empty. So another actually base is attached on that side, and once the base is attached, you are actually making a bond with the sugar group there, right? So because sugar is a backbone. So once phosphate actually binds with the 3', what happens? It releases, because previously there was an OH there at the 3' place, and when O binds with the phosphate PO4, the O binds with the OH, actually it removes one hydrogen. So the hydrogen is a byproduct of the chemical interaction, and this is how it works. So whenever you have you're elongating the DNA, hydrogen is produced for each base. So when hydrogen is produced, whenever in a solution, if you have more hydrogen ions, what does it mean? More acidic, right? pH. So it means the pH will be low. Yes. So with each addition of a base, you will have a pH change in the solution. Now think about if there's only on a bead, there's only one DNA strand, and then your base is attaching, so you will have one hydrogen produced. Can you sense the one hydrogen in small? No, one hydrogen will be just impossible to sense in this kind of a setup. Okay, because you have maybe 10,000 other hydrogen ions also will. But now let's say if you have 1 million hydrogen produced, then you would be able to, that will significantly change the pH of a solution, and you would be able to sense that. How we can sense the pH change? One way is how they did it is actually by using the CMOS transistors, MOSFETs. And we did talk about briefly about the MOSFET in this class, right? Source, drain, and gate regions. Yes or no? Yes. So then it might have been recorded, so you can look at it. But briefly, let me show you once again. So hydrogen, which is H plus, is getting produced. So in transistor, we call it a bio-FET because of field effect transistor, and bio because it's used for bio applications. But it's a similar transistor. So in regular transistor, which is found in a computer and other places, electronics. So you have actually a gate here. This place is called gate, which is controlling. Then you have okay, so this is a source, this is a drain, for example. And then you apply voltage. Between these two terminals, and this is a semiconductor, okay? This place is semiconductor, silicon. And these are doped regions, so this is mostly N-type or P-type or N-type based on whether it's NMOS or PMOS. So let's say in this configuration, if you apply some voltage, let's say plus 1 volt, will current flow through it? Will you see any flow of current if I apply voltage? So current can only flow from this part to that part to complete the circuit, and it will depend on the conductance of this channel. And this is a semiconductor, I told you silicon. If it's silicon, it's a semiconductor, it will not allow the current to pass unless you really apply high voltage. There will be might be leakage current in picoamperes a little bit, but it's considered as no current is passing through. In this case, there is no current because there are no mobile carriers here. Okay? And then you apply some voltage at the gate. Let's say now you apply some voltage between gate and source. To make it simple, I'm just showing you that apply voltage here. You apply some plus voltage here, for example. Now if you apply some voltage, and because of a capacitive effect, what happens? It will pull electrons close to the surface. Right? It will pull electrons close to the surface. Because there were some electrons there in a semiconductor and also some holes, but they are very little amount. And now with this application, we are pulling all of them close to the surface. So it means now this area will be a little bit more conductive as compared to the rest of the area, because now you're pulling those electrons there with the charge. And now when you apply voltage, the current will start flowing through the channel. You will see current flow. And that's how a transistor works, this transistor. So with the gate, we can control it. Now think about, instead of applying voltage at the gate, if there is more hydrogen, H+, 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 and we expose the gate region to the solution which has more hydrogen ions, so it means the gate will face more hydrogen ions, so that will pull electrons from the bottom, because that area will not be conducting water. Only the gate is conducting water. So the hydrogen ions will come close to the surface, it will bring some electron on the top, and you will see current flow. So it means when you get a current flow, that is dependent on the concentration of the hydrogen ions or a pH change. So the hydrogen, now if you look at it, so you have a drain source, and this is actually your, in here it says a sensor plate, but actually it's a gate. Okay, and whenever there is a hydrogen, so it changed the charge, and because of that there is a capacitive effect, and you'll see some electrons are getting pulled. This is how transistor works in a computer also. And then you will see a current flow, and when you see current flow, it means there is something going on there. So you can sense, this is actually, in brief, it's actually a pH sensor, so you're sensing the concentration of hydrogen ions. Now we push A, it will bind different places where there is a T, and you will have only pH change. You'll sense the pH change very quickly. So that's the power of technology now, because now you can even control millions of transistors on one chip very quickly, and you can address all of them. This is how computer is designed, and that's why you have a lot of transistors in the microprocessor. So addressing them, sensing them, we already have technologies in place. So it was not something new, but the application was new. So the technology was already there where we can control many different things electronically, and that's the power when we choose our design based on something which is already very well developed. So in this case, they were able to read million different wells very quickly. Okay, each place they are sensing, okay, where there is a pH change, this well, that well, this well. So it means you have a T there. If there are two T's, then you will be two hydrogen ions, pH change will be more, you will sense that also. So then you just wash it, and now flow another base, which is a T or G or C. So in that way you continue this process, and then again repeat the first one, A, then T, G, C, then A, T, G, C. But in this case you don't have to remove the dye, so you can skip that step only. But otherwise the process itself is very similar how the Illumina platform works, but the sensing mechanism is different. So, and instead of using fluorescence, you use electrical sensing, and it's a label free, it doesn't require any labels. And again you can sequence very long DNAs, breaking them into small segments, and with this process. Now there's another technology, it's called Nanopore, which we have talked briefly a couple of times previously, and that's also being developed for DNA sequencing and also protein analysis and a lot of other applications. Are mostly the DNA idea was to develop it for label-free DNA sequencer, and this company actually started at 2011 or 10, I remember, because when it started, I looked at it at that time, and they started developing it. There are a lot of other people who also developed, who also worked on nanopores and different aspects of nanopores. But this company was the one who started to commercialize. One of the first companies who started to make a commercial product, because I think in 2000, last 10-15 years, you will see thousands of articles and research groups. Many research groups are working on the nanopores, but there was no commercial technology out there because the system is not very reliable. I work with this system myself, and how it works is you don't get a signal all the time. So they put a team to actually fine-tune those things, and so the idea of and now they have now commercial product available, which people can buy, and they call it a MinION. So they are this size of a device where you can insert a pore, and there is a way to follow. So there is a protocol to follow the process, but you can end up actually sequence the DNA. It has some high error rate as compared to Illumina right now, but they are improving also on that. How this works first. So in Nanopore, the working principle is very easy. It's a electrophoretic mobility of a DNA. In this case, minus, plus. So your DNA here, it will pass through the pore. So once it's passing through, you'll get some dip in the current. And now the idea is, I told you that we can, based on how much deep it is, we can, how much length, how much is the translocation time. This time is called the translocation time. That's the time it takes to actually pass through the pore, and from there we can estimate, okay, how much is the length of a DNA, okay, with the help of a control, and you can also control that with the how much voltage you apply. So in here there is no gel, and this is all filled with electrolyte, KCl, NaCl. You can just make electrolyte and let it work like that. So the goal was that once the DNA passes through, so because each base have different functional groups, is chemically slightly different than the other, so we may end up actually getting pulses which can be distinguishable based on the bases. That was the idea. So if you look at this example, just to show, this is just an idea, like when the DNA is passing through, so you might get some pulses which are deep, you might get which are longer. So the idea was that might show that we can look at we can actually trace those peaks or the pulses back into the different bases. So A will produce slightly different base, different pulse shape, and the T will produce slightly different. So in this way, you can let the DNA to pass through, and based on those bases and pulses, you may be able to sequence the DNA. So that was the idea. And now based on how it works, you can think about bases are very close to one another, and to sense that, it will be a very hard job. And it is a hard job. But they managed to actually overcome some of the limitations, and there are ways to do it. So one method was that you can put an enzyme at the inlet of a DNA, and as long DNA is coming by, so the enzyme will cut base by base. So then each time only one base will pass through the DNA, through the nanopore, and then you can get a distinctive signal if we have the resolution that we can differentiate those pulses based on the chemical nature of the bases. So we should be able to actually find out the different pulses. So, but chopping them, then again, you can chop it individually, but then whether that base which you are chopping will passing through first, then the second one which you are chopping, there's still there'll be a lot of noise. So and there are now the reason is, if you look at the nanopore, although we make a very thin membrane, if you look at here, so we make a very thin membrane, and but still it's thick. So thin membrane means 15 nanometer, let's say, very thin, 15 nanometer thin, and that means from here to here is a 15 nanometer. Oh, sorry. Okay, you're not looking at it. Thank you for letting me know. Okay, maybe that's why. Okay, so I was talking about. Okay, let me just repeat part of it. I think I was thinking that you were saying it, so that's why I was explaining it from here. So the idea was that you can get these pulses, and the shape of the pulse can reflect back or the base which is passing through. And in this case, let's say they were saying this is a T, then you'll get a different pulse, and if there's a G, you might get a different shape of a pulse, and then there was a goal. Okay, these are not real pulses; it's just a drawing. And now in this case, when the DNA is passing through the pore from negative to positive side, then this image, before I continue there, this is the AFM image, one from my papers, and it took us a long time actually to take this image. This nanopore is about 10 nanometers, and the colors are, we just gave them fake colors, so it looks very nice. And so it's a 10 nanometer, and it's an AFM image. So look at you know how AFM works, right? So it has a very high resolution. Okay, so the DNA can pass through the pore, and as passing through, you can think about now, as I was telling you, the membrane thickness will be 15 nanometer. Let's say it's a 15 nanometer. How many DNA, once it's passing through, will be inside a pore when the membrane is 15 nanometer? So 15 nanometer means 10 bases is 3.4 nanometer, right? So 15 divided by 10 divided by 3.4. Let's say 3.14, 15, it's like 5. So it's about 50 bases, right? Correct? 10 into 5, yeah, about 50 bases. So 50 bases would be inside the So about 50 bases will be inside the DNA at one time, inside the nanopore at one time. No? 10 bases equal to 3 nanometer, 3.14.
Okay, yeah.
Right?
Yeah.
So it will be 50 in case if the membrane thickness is 15 nanometer. So it means at one time it's 50. So how we can differentiate if at one time we are getting a signal from 50 different bases? So once the DNA passing through, I don't know how we can differentiate if the signal is because of some specific base or not, right? So it's difficult, and that was a challenge, big challenge. Okay, we'll see what are the possibilities, how we can fix it, and that's why they put some enzyme to cut it base by base, so that at least we can isolate bases from one another. So at one time only one base will pass through, not 50. Okay, and there is also interest to make membrane more and more thin. If you make it more and more thin, it will break also very easily. And those are challenges actually. And then DNA when passing through, it doesn't pass very slowly, it passes very quickly. Zoom, you apply voltage, it passes very quickly through the pore. If you reduce the voltage, you can control the movement to some extent, but still you need some voltage. Still it passes very quickly. So then we don't have a system which can read the signals so quickly from after in a few milliseconds, few microseconds. You have to get a signal from there, and you cannot get a signal from single base. So that's a challenge. That was a challenge. Okay, before I continue with the how we can fix some of those issues, how those issues fixed, I want to give you a little bit more background about the nanopores. So the nanopores, the idea of nanopore doesn't came from nowhere. We already have pores in cells. You might have heard about ion channels, right? So ion channels which can regulate the potassium and sodium ions inside the cell and outside the cell. If we watch a video like biology life of something about how the cell membrane and the cell itself works, in that time you might also have seen there are some channels which control the movement of the ions, and those are gated channels. It means you can control the movement. That doesn't allow free movement, so there is a control how much ions can go inside and outside the cell. So it means those ion channels are actually also pores, right? The pore is the small opening through which the ions can pass, and the ion channel is also exactly similar. So it's a very small opening where you can control the movement of the ions from one side to the other side. And there are pores in a nucleus, cell nucleus, which allows the RNA to pass through, or DNA to pass through from your chromosome to the in the cytoplasm, so that the proteins can be synthesized accordingly. So there are pores in a natural environment, and there is one biological pore which is very well known. It's called the alpha hemolysine. It's an ion channel which is present naturally, and that ion channel has a alpha hemolysine based protein ion channel. It's a protein, it's not a solid state. It has a pore diameter of 2.6 nanometers. So when you assemble that ion channel, and you can assemble it outside, you can take those proteins, mix them at certain ratios, and they will make the channel like this. This is a protein channel. For that you also have to make a lipid bilayer, which is not difficult to synthesize on device. So you can first synthesize the lipid bilayer with the phospholipid ions, and then you insert this channel, and there is a way to do it. So a lot of people have demonstrated it. So you can actually make this kind of a channel, single channel, outside in a device. And now this channel, if you put it in a chemical cell where the DNA can pass through, the size is very good. The good thing about the biological nanopores is that you will not worry about the size. Always you get 2.6 nanometer. In a solid state, means the devices which we make on wafers, the size, although we have very good control on the size, about 2 nanometer, but it can be 2.5, it can be 3 nanometer, right? So plus minus 1 nanometer difference is very easy to happen. So we don't have that perfect control when we are making those pores with the TE transmission electron microscope or focus ion beam. But with this one, we have perfect control. So that's why a lot of people are still working on the biological nanopores, and they're looking into other protein channels which can be used for this purpose. The good thing is that we always get 2.6 nanometer, but the limitation is that if we want to change the pore size for a certain reason, we cannot do it in this case. Because it's a 2.6, it will remain 2.6. So maybe we have to then find out some other ion channel which have larger pore size or smaller based on the application. So we don't have much control on changing the parameters of the pore itself, but we always get a very consistent pore. And these pores, we can also make it in devices, right? In a membrane, you already know that there was also one midterm exam question. So, and we can make these pores in a membranes, and then we can put it in a chemical cell. And so the goal is that once the DNA is passing through, you get some pulses. So people have shown, so you can look at, for example, this example. These are some of the examples. For example, in this paper, they were able to discriminate 30 base pair long DNA fragment, one with the 1 base pair difference. Means there are two 30 base pair types fragments. One has a 1 base pair which is different from the other, and then when they run it, they notice that where there was a perfect complementary, they already attach it inside a pore. Let's say if we have this pore, so we already attach one single strand DNA there, and then let the other single strand to pass through. So once you pass it through, when it was a perfect complementary, they were seeing that that binding, because it will try to bind with that, the binding was showing very typical, very different pulse as compared to when it was not perfect complementary. Then it was a little bit more quicker, and the pulse shape was different. So they were able to distinguish between two 30 base pair long DNA fragments, which was just one base pair. There was one base which was different, but so it means we can distinguish two fragments of same size which have some changes, but we cannot really still, after this is a significant development, but we cannot still sequence each base so far. But I told you that there are already technology there. They were sequencing it, but I'm just taking you where it was before. So this was the first, I think, report which shows that they can distinguish the DNAs from with only one base pair difference. Okay. And now there are other types which are solid state nanopores. I think I will stop there. Okay, and then because it will continue into other topics, so I'll stop there, and then I'll see you next week, then on Tuesday, and we'll continue from this point.